I am getting here. Probably the kindly confirm that the students are attached and मेरी आवाज़ अगर जा रही हो तो confirm करें because the network is not informed that the students are connected, so it is necessary to confirm by the message. Some students are attached. Okay, now I am initiating your live class or live lecture. Now, today, the topic which I proposed for the discussion is metabolic blockers. Of course, the metabolic blockers is important topic for the MSc level, and it is also most important for the CSIR and NET examinations, because in each examination at least one or two questions are asked from the metabolic blockers. So the large numbers of metabolic blockers are reported, and today we are going to discuss on the metabolic blockers in the form of antibiotics or the chemicals responsible for the blocking of energy system. And before coming on the topic, it is necessary to keep some fundamental concepts for the blocker. And what is blocker? So at first, actually, in the first concept of the antibiosis, the term used for the bacterial inhibition, and the antibiosis was at first the concept of the antibiosis, not the term of the antibiosis. The concept of the antibiosis was at first presented by Posture and Koch, and he reported that uh, airborne bacillus, that is a bacteria, airborne bacillus, is responsible for the inhibition of the growth of Bacillus anthracus. And he proposed that the role of the airborne bacilli is responsible for the retardation of the growth of the Bacillus anthracus. And in this way, he at first suggested a concept like the antibiosis. Antibiosis means that the one organism is responsible for the retardation of the growth of the organisms present in surrounding. And now it is widely used in the lower plants or plant sizes and in the microbial populations. So the concept of antibiosis was at first suggested, but the term antibiosis was coined by Woolman. At first, the term coined by the Woolman, and on the basis of that form or that action, the first antibiotic was reported by Alexander Fleming in the form of penicillin in probably in 1920s, and that penicillin was widely used during the treatment of the in the treatment of the wound during the First World War. And for that work, Alexander Fleming, in association with the two other workers, awarded Nobel Prize because the penicillin is extracted in the pure form by some other workers. And now I am not going more detail on that particular part, but the antibiotic, first reported antibiotic was the penicillin, and that penicillin was at first reported from the microbial population or the microorganism. Now, the one thing is more that the term antibiotic is uh, proposed or coined by the Waxman, and Waxman is also responsible for the reporting of the streptomycin, and that streptomycin is widely used and in the treatment of the tuberculosis, and for that purpose the Waxman also obtained the Nobel Prize. So here some fundamentals which I am discussed here. So it is necessary to clear the concept that the metabolites either produced by the microorganisms or by the um, chemically synthesized metabolites, which has if negative impact on the growth of the microbial populations or other organisms, are responsible for the action by inhibiting some molecular or metabolic activities. So, today we are confined our lecture or our study to the energy prohibiting or energy the metabolites responsible for, or the chemicals responsible for the blockage of the energy production or blockage of the molecular activity. And for that purpose, the antibiotics are used for the treatment of the diseases. So here, one or two things are necessary before coming on the blockage technique. If we use the chemical, so a large number of antibiotics are extracted from the microbial populations, but the all chemicals or antibiotics produced by the microbial populations are not suitable for the treatment, because if the antibiotic synthesized or extracted from any microbial population has equally negative impact on both prokaryotic and eukaryotic cells, then it is very difficult to use it for the treatment, because it is also responsible for the blockage of the molecular activities of the prokaryotic cell as well as the eukaryotic cells both. In this condition, the first important or significant feature which is necessary for the use of any antibiotic for the treatment is its specificity. So, the specificity to the prokaryotic molecular activity is primary factor for the use of any antibiotic synthesized for the microbial population in the treatment. And so it is well known that the naturally all antibiotics are synthesized and released by the microbial population. However, some synthetic antibiotics or secondary modified antibiotics are also used for the treatment, which we are not discussing today, because I am confining the parts of the information which are necessary for the for your NET and CSIR examinations. So at first, you it is necessary to understand one thing, that is the specificity to the prokaryotic cells. So And second one, that is we use the antibiotics either as a bactericidal chemical or as the bacteriostatic chemical. What is the bactericidal chemical? That is bactericidal chemical which is responsible for the killing of the bacteria or microbial population. And if any microbial population is released any toxins after the killing, so in that condition we use the bacteriostatic antibiotics. So the two types of antibiotics are present: bactericidal and bacteriostatic. Bacteriostatic antibiotics are responsible for the lowering of the growth rate to the sub lethal conditions, or the conditions which is not responsible for the any pathogenic damage to the body. So they are the part of the treatment which I am just describing in brief. Now we come on the topic of the blocking. What is the action of the different types of antibiotics? But because sometimes the questions are asked in the NET and CSIR examinations, and asked directly that what is the action of the antibiotic, and you have the four or five chemicals, four options are presented. That four actions represented the four different mode of the blocking as proposed in the questions, and question asks that you select a one, or sometimes one or two parts are also questions are also presented in the form of the one or two options. So it is necessary to understand the blocking nature, point of the blocking nature for the antibiotics. And if we observe, or if you study the microbial population, then it is the chemical feature that the microorganisms have outer cell wall, and inside the cell wall genetic material is present, which is responsible for the mm, transcription and translation. That transcription and translation are necessary for the growth, and now the metabolites. So in the bacteria, the point of blocking is first the cell wall. If any blockers blocks the cell wall synthesis or the formation of the cell wall, then the growth of bacteria is retarded. And if the cell wall is not formed, that bacteria is easily lysed by the immune system of the body. So one part is the cell wall, one point of the blockage is the cell wall. Second, the protein synthesis. Fourth is the transcription. Third is the transcription. Fourth is the replication, and fifth is the metabolite. That means the blocker can act on these 5 points. So it is necessary to remember that which type of antibiotic is responsible for the blocking at which level, and what is the point which is blocked by the antibiotic. So we are just going to discuss on the first part, that is the blocking of the cell wall. The cell wall, if we study that the cell wall, and the cell wall, if we study the bacteria, we easily observe that the bacteria is of two types, that is the gram positive and gram negative bacteria. In the gram uh, positive bacteria and the gram negative bacteria is differentiated. बंद करो. The gram positive bacteria and gram negative bacteria is differentiated on the basis of the state. That means in the gram positive bacteria, the gram stain is responsible for the staining of cell wall. In gram negative bacteria, the cell wall is not stained by the gram stain. Here, the gram positive bacteria is associated with the stain by peptidoglycan. So, the peptidoglycan and thiazidelic acid are prominent components of the structural components of the gram positive bacteria. But in gram negative bacteria, in addition with the peptidoglycan and thiazidelic acid, three extra layers are present above it. So the wall of the gram negative bacteria is more strong than the gram positive bacteria. And here the two antibiotics, that is the penicillin and cephalosporin. Penicillin and cephalosporin are uh, responsible for the blockage of the synthesis of peptidoglycan. So, it is responsible for the blockage of the cell wall synthesis and in easily in the gram negative bacteria, gram positive bacteria. In gram positive bacteria, the cell wall synthesis is blocked by the penicillin and cephalosporin, and both the antibiotics belong to beta lactam group. That is the chemical nature of the cephalosporin and penicillin. Now, the number of beta lactam antibiotics are synthesized chemically, but it is the natural, penicillin is the natural, and why so it was widely used during the wound healing and wound treatment of the First World War. Now, the second one is the polymaxin. Second one is the polymaxin. And polymaxin is responsible for the um, blockage of lipid A, blockage of lipid A, and it is active in the gram negative bacteria because it enters, it crosses the outer wall, and in the inner wall it interferes with the lipid A, and it stops the or it activates the disruption of the outer membrane. So the polymaxin is responsible for the synthesis of the cell wall in the gram negative bacteria, while the beta lactam antibiotics like penicillin and cephalosporin are responsible for the prevention of the or interference in the synthesis of the peptidoglycan, and then they are responsible for the blocking of the synthesis of cell wall. Now, the uh, second stage is the action of antibiotic at transcription level. And at the transcription level, the most important chemical, and which is generally asked in the examination, is the actinomycin. And actinomycin binds at site rich in the GC bases of DNA, and after the binding, it blocks DNA to RNA. That is the RNA polymerase responsible for the transcription from DNA, and after the binding to the GC rich site of the DNA, actinomycin prevents the action of RNA polymerase, which is responsible for the transcription and synthesis of mm, messenger RNA, and that means it is responsible for the prevention of the synthesis of protein. So the protein synthesis is blocked by actinomycin D, or commonly it is referred as actinomycin D. It is responsible for the blocks the RNA polymerase, and in higher concentration, if we use the higher concentration, it is responsible for the disruption of the DNA. So here the two activities are observed for the actinomycin D. And so, when question is asked in the NET and CSIR examination, it is necessary to understand properly that which type of question is asked, or if both are represented, then you select the answer accordingly. In the higher concentration, the actinomycin D is responsible for the blockage of, sorry, disruption of DNA, and that disruption of DNA is prevented the replication. If they are disrupted, if the DNA sequences are different, dis, disestablished, then there is no chance of the replication. Why so? In on this principle, the actinomycin D is used in cancer treatment due to its ability to inhibit transcription and disrupt the growth of division of the cancerous cell. So sometimes the actinomycin D is used for the um, cancer treatment. Now, the next stage where the antibiotics is acted is at the first, you consider in the prokaryote, the protein is the ribosome, which is responsible for the, which is associated with the protein synthesis, as 70S, and 70S is dissociated into the 30S and 40S, the two subunits, sorry, 30S and 50S, that the two subunits are associated to form the 70S ribosome. And here the ribosome is actually formed by the ribosomal RNA and proteins, and it is associated with the mRNA to form the protein initiation complex. Just I am saying that you properly understand what I am going to say. And here, the first antibiotics, we which is responsible for the blockage of the 30S subunit, and the 30S ribosomal subunit, the antibiotics which act on the 30S ribosomal subunit is streptomycin, gentamicin, and kanamycin. In this way, all the aminoglycosides, all the antibiotic based on the chemically known as the aminoglycosides, are responsible for the activities of 30S ribosomal subunit. So you remember that the streptomycin, gentamicin, kanamycin, these are, or the all groups belong to the actinoglycosides, are responsible for the blockage of the 30S subunit. Next, the antibiotics blocks the 50S activities and elongation of the polypeptide chain. That is the erythromycin, azithromycin, and all the mm, group belong to the claridomycin, and this group is responsible for the blockage of 50s ribosomal activities and prevent the elongation of the polypeptide chain. That means it blocks the elongation of the polypeptide chain by acting on the 50s ribosomal subunit. Now, the uh, two antibiotics, that is tetracycline and doxycycline, both acts on the 30th. I am just saying that both act on the 30th. But the blockage is different as for the streptomycin. So it is necessary to remember the what is the difference, because the tetracycline and doxycycline act on the 30S. But what is the difference? The tetracycline and doxycycline prevent the binding of amino acyl tRNA, because the tRNA is acylated before the attachment to the initiation complex. The ribosome complex is formed, and before the formation of the ribosomal complex, the tRNA is acylated, and then the acylated tRNA comes at the site and associated with the ribosomal, and ribosome has the A and P sites. As I think that you all know these fundamentals concept. And here the tetracycline and doxycycline prevents the binding of the amino acyl tRNA to a site. And when it is not associated with the A site, then there is no chance of the further formation of the bond and elongation of the chain. So here the mode of action of the tetracycline and doxycycline is different. Now the next is the chloromystin, that is chloramphenicol. That is, chloromystin is chemically known as the chloramphenicol, and it is generally asked in the your examinations that what is the mode of action of the chloramphenicol, lincosamide or cladid and clendocycline. These are the synthetic actions, but it is better to you remember the chloromystin or chloramphenicol. And the chloramphenicol is responsible for the prevention of the activity of enzyme mm, mm, peptidyl transferase. And that if the peptidyl transferase is blocked by the chloramphenicol, then there is no chance of the formation of the peptide bond between the carboxyl group of the first amino acid and amino group of the second amino acid. And in this condition, there is no chance of the further elongation of the chain. Now, the another blocker, that is the oxalo oxalolidicine and and linositol components, and they are responsible for the blockage of the activity of 23S RNA of 50S 50S ribosomal subunit, because the 23S RNA 23S rRNA is essential structural component of the 50S subunit, and this group of the oxalolidinones and the linezolid both are responsible for the blockage of the action, and in this way it is responsible for the initiation of the synthesis. Because it is blocked, then there is no chance of the initiation. Then the other blockers, which is responsible for the blockage of metabolite. We commonly know that some metabolites, and one thing is important, that is folic acid, which is asked sometimes. The folic acid. And the folic acid is the metabolite which is responsible for the, which is associated with, which is necessary for the synthesis of the DNA and RNA. The biosynthesis of the DNA and RNA in bacterial cells is associated with the presence of the folic acid. It requires the folic acid. And the some antibiotics, that is the containing sulfa, sulfa groups, that is the sulfa amidines or sulfamides, blocks the activity of the folic acid. And so, the if the folic acid is blocked by the antibiotics, then there is no chance of the synthesis of the DNA. And when the DNA is not synthesized, then there is no chance of the growth of the bacteria further. So, in this way, these antibiotics are used as a blocker for the metabolic activities, and they are responsible for the retardation of the bacterial growth or killing of the bacteria. Now, the second part, which is generally asked in the your NET and CSIR examination, is the site for the blockage in the energy pathway. That is the in the ETS, electron transport system. Here, some questions are also asked. Sometimes, or sometimes in one or after alternate examinations, the question is asked from there. And in uh, one or before 1 and 2 class, I explained the ETS, electron transport system. And in that electron transport system, I was said that the sequence of the electron transport chain, that is ubiquinone, after the ubiquinone, the flavoprotein, the flavoprotein, ubiquinone or coenzyme Q, after that the flavoprotein, from flavoprotein the cytochrome B, then the cytochrome C1 and C2, then the cytochrome A1 plus A3. And in this way, in that time when I was explained, and just I am again repeating here in brief for the proper understanding of the blocking, I said that in the electron transport system, the coenzyme Q is responsible for the receiving of H plus from the NADP or NADPH2, whereas flavin is responsible for the receiving of the H plus from the FADH2. And after that, the electron is excited, and electronic movement is responsible for the creation of potential gradient, and they pump the H plus. And when the electron is moved from the higher energetic level to the lower energetic level, it pumped the H plus, and that H plus gradient is responsible for the conversion of ADP into the ATP, and finally the electron or H plus is neutralized by the oxygen, and it forms the H2. So the site of the H plus pumped out, and the blocker which is responsible for the blockage of the site of the electron transport chain is necessary, because sometimes the blocker action is directly asking the question, and sometimes the ATP is asked that suppose anyone say that the X blocker is used, and after the use of the X blocker, how much ATP is produced by the electron transport system. So in this condition, two informations are simultaneously required. One, then at what is the point where the H plus is released sufficiently for the synthesis of the ATP, and which point is blocked by the particular blocker which is asked in your question. And if we know both accurately, then you will reply the correct answer. So, in the electron transport system, I clearly mentioned that what is the point for the synthesis of the ATP. And here I am going to explain, or I am going to inform you that which type of chemical is responsible for the blocking at which site of the electron transport system. So both are facts are necessary to remember and keep always in mind. So I am just here saying that which type of chemicals and what is the point of the blocker system. First chemical which is responsible for the blocking of the electron transport system is rotenone. First is the rotenone, and rotenone is responsible for the blocking of transfer of the electron or creation of the H plus gradient at complex coenzyme A to flavoprotein level. The coenzyme A transferred the component to the flavoprotein, and rotenone is responsible for the blockage at this level. So there is no chance of the production of the ATP when we use the rotenone, because it blocks the transfer of the coenzyme A to flavoprotein. And here one ATP gradient is formed, and if the any step is blocked after the flavoprotein, then there is chance of the production of the 1 ATP, because 1 ATP is produced at the site when electron is transferred from coenzyme A to flavoprotein. Why so? The FAD is responsible for the production of 2 ATP, and NADH is responsible for the production of 3 ATP. So here there is no chance of the production of the energy or ATP. Second one is antimycin, and antimycin is responsible for transfer of electron from cytochrome B to cytochrome C1. You remember it, that antimycin is responsible for blockage of electron transport from cytochrome B to cytochrome C1. Next one is the cyanamide or CO, carbon monoxide, which is commonly asked in the your net examination. Cyanamide and CO. And cyanamide and CO is responsible for the blockage of cytochrome oxidase activity at A1 plus A3 level. Now the next chemical is the oligomycin, and oligomycin acts as the blocker of ATP synthetase. So oligomycin is also Here it is not directly involved in the uh, ETS or electron transport system, but it is responsible for the blockage of the enzymatic activity of the ATP synthetase. So the gradient conversion, the electrochemical gradient is created, the potential change is created, that is sufficiently created for the synthesis of the high energy high energy group and synthesis of the ADP to ATP. But here the ATPase enzyme is not produced. In this condition, it is very difficult to synthesize. Now next is uncouplers. Some compounds are known as uncouplers, and the uncouplers dissipate protein gradient. It is dispersed the protein gradient. Protein gradient is created, but that protein gradient is not used for the synthesis of the ATP from the ADP. That is referred as the couplers. And some important couplers are 2,4-dinitrophenol. It is the coupler. Now the salicylates. It is the coupler. Then phenol derivative, 100 phenol, and these all these components are referred as couplers. So the couplers are responsible for the dissipate the potential synthesized. So these are the common metabolites and commonly asked in your NET and CSR examinations. So now today I am just describing the blockers which are necessary, which are important for the competitive examinations, and sometimes one and two questions are also asked in your neat some simple questions. So now I am thankful to all viewers or all the students attached today, and just after 1 or 2 days, I am again suggested you the new topic, and probably because the classes are not going now, so we use the live class for your further discussion. Now, thank you to all, and if you have any question or anyone has any question, then give me message. I am just waiting for 1 minute. Probably no one asked question. So thank you to all. And it is my kind suggestion that it is the most important topic for your NET and CSIR examination, and regularly asked question from this part. Thank you. Now good night.